Yeah. Alessia Gallerani, yeah, you can take a seat. Andrea Masetti, can take a seat. Giulia Oddi, very good. Ian Regueiro. Oh, Ian, how do you pronounce your name? Ian. I'm sure that by now you know the rules of the game. So would you like to have 3 or 4 minutes? Have you timed yourselves? So if you're sure that you will not exceed 4 minutes, then I will relax. 4 minutes at, 4 minutes at the latest, okay? Good. So this is how we're gonna do this. Each and every one of you will present the pitch, and then we'll have a discussion with the 4 of you and the audience, right? Good. So first one is you. Let me go back to the list. Yep. Alessia. Good. Your turn.
So good morning, everyone. I'm Alessia, a PhD student in ICT at the University of Modena, Reggio Emilia. And today I'm going to show you my research topic, which is about the development of sensors for biomedical applications. First, let's see where our laboratory is located. It is in Mirandola, in the heart of the biomedical district in Emilia-Romagna, which is a very important area because here we have a very high production of medical devices, in particular oxygenators, that are very important in clinical contexts. For example, during extracorporeal circulation, in which they help to oxygenate blood. And during these procedures, it is fundamental to keep track of the variations of blood parameters that are called critical care analytes. And in my research, we focus on the detection of blood partial pressure of carbon dioxide. And in a clinical context right now, there are no evidence of the use of devices that are able to monitor in real time in a continuous way and in line with this circulation circuit. And so what we wanted to do is to develop a prototype of a sensor that is able to do these 3 things at the same time. And we wanted to exploit the hollow fibers technology, which is the same on which oxygenators are based. These fibers are made of polypropylene, are hydrophobic, porous, completely safe for the patient, and we can totally produce them in our laboratory. So essentially what we were able to do is to develop a prototype of a fluorescent sensor for blood partial pressure of carbon dioxide, and it is composed by 2 parts. The first, that is the one that you see on the left in the 4th square, is the disposable part. It is the sensitive part in which we have the fibers, blood flows inside them, and it can exchange CO2 through the pores. And this CO2 is absorbed by a pH-sensitive fluorimetric solution. And this CO2 changes the pH and the fluorescent response. And we can detect this variation with the second part of the sensor, that is an optical head. It is the reading device, it is fixed, and it is based on an optoelectronic technology. And thanks to this, we are able to detect these variations, and through some mathematical models, we are able to estimate blood partial pressure of carbon dioxide. We also tested it with blood, and it gave us pretty good performances. And we are still developing this kind of device, but we are confident for the results. So this is more or less what I'm doing right now. And if you want to contact me, there are my contacts. And thank you.
Thank you. Very good, very clean and clear. You've used your time very wisely. Up next is Andrea.
Okay, so hello everyone. I'm Andrea Masetti. I'm a PhD student in industrial chemistry at the University of Bologna. And on a daily basis, I deal with chemistry for plants, or more specifically, what chemistry could do to help them thrive and prosper. Now, there are multiple challenges nowadays that we have to face from an agricultural point of view when dealing with plants. And one of the main one is the iron deficiency. Despite iron being one of the most abundant elements on Earth, actually ranking as the fourth most abundant element if we have a look at the Earth crust, in alkaline soils, which comprise about 70% of the world land population, iron is mostly available in forms that cannot be uptaken by plants. This leads to the rise of a very common disease, which is known as iron chlorosis, which elicits, from a visual point of view, as you can see from the slide, the yellowing of the leaves, but most importantly, from an agricultural point of view, a huge reduction in crops and fruit yield down to minus 80%. So it's a huge problem. Now, the solution that we apply as humans nowadays in the industry is the application of iron chelates, which are molecules that are highly polluting to produce and they are not biodegradable. Therefore, they do remediate the iron chlorosis, but they do stay in the ground and cause pollution. And this is where my project started. In a world that leans towards sustainability, I found that these agricultural techniques are not feasible anymore. So I wanted to find a new solution. And together with Professor Namba from Osaka University, we decided to take inspiration by nature. Now, gramineous plants, such as rice, synthesize and produce these small molecules known as homogeneic acids, which are molecules that facilitate the absorption of iron even in these unsuitable conditions. However, this homogeneic acids are just a survival mechanism, meaning that they just synthesize the bare minimum to survive, but they're not going to be able to overcome iron chlorosis. But these molecules have a high, very good affinity for iron, so we said, could we make them in the lab? These molecules are biocompatible, are biodegradable, and could we bring them to be a fertilizer? This is where our project started. So we were able to develop the full synthetic pathway to obtain 2-deoxyhomogeneic acid, which is a molecule produced by barley, as you can see, but we encountered two main problems. First of all, a problem of stability. The molecule tends to decompose after 24 hours, which is not good, and a very high production cost. This is where I stepped in, and I decided to play a little bit with chemistry. I wanted not to have the same molecule, but something similar to 2-deoxyhomogeneic acid. So I tuned a little bit the left end of the chain, and I changed the 4-member ring with a 5-member ring, which allowed me to increase the stability up to 40 days, so probably, and a cut down of cost in the power of 100. But I was not satisfied yet. So I tuned also the right end of the molecule. I moved one chiral center, and I inserted some easier, let's say, chemical substances. As you can see from the slide, so far I was able to obtain 7 analogs, 1 to 7. But of course, no fertilizer is any good if it doesn't have any biological activity. So we did some biological testing together with Aichi Steel, a company from Japan, on rice sampling and daikon sprouts. And the most interesting thing, is that these molecules have a very good remediation activity on iron chlorosis, even compared to homogeneic acid, but most importantly, when compared to the available commercial fertilizer nowadays in the market. So for now, I'm going to stop here. This is my research so far, but I'll be here, so if you have any questions, please be free to ask, and thank you for the attention.
Very good. Excellent presentation. Very good. Thank you. Giulia Oddi.
Good morning, everyone. My name is Giulia Oddi, and I am a PhD student at the University of Parma in Information Technologies. In particular, my research mainly focuses on smart agriculture, and so in the application of Internet of Things technologies and also artificial intelligence algorithms into the agricultural sector. In general, the main objective is to optimize traditional farming practices in order to create a more sustainable and efficient agriculture. With my research group, we implemented a general data acquisition platform that is called Agriware. This platform is useful to collect, monitor, and analyze data coming from different and heterogeneous sources of information within the smart agriculture context. As an example, as you can see in the slide, in the last 3 years, we managed an evolving tomato test bed located at the Azienda Agricola Sperimentale Steward in Parma. And in this test bed, we positioned IoT network of different types of sensors in order to monitor the general condition of the crop. Moreover, with the AgriWare, we were aware to monitor the behavior of plants under some water stress. And in the last experimentation of this year, we implemented also some algorithms for the generation of an automatic irrigation process. Another line of my research is based on hop cultivations. In this case, I've implemented an algorithm capable of predict the optimal harvesting time for hops plant. And right now, we are working with the implementation of some artificial intelligence algorithm capable to identify and classify the sex of plants. In fact, hops are dioecious plants, and this means that the plants can be male or female. And finally, another ongoing research is about the monitoring of the growth of basil plant. In this case, we implemented a network of different sensors and cameras in order to monitor the growth of the plants subjected to different water regime and also to different biostimulating treatments. So my work, my research focuses on the application of information technologies in agriculture. And that's it. Thank you all for the attention.
Konnichiwa. My name is Ian Regueiro. I'm a PhD student in the University of Ferrara in environmental sustainability and well-being. So today I'm going to talk about archaeogaming, that is simply archaeology and video gaming, is my research. And here you can see my game loop. So my methodology and what I'm proposing with my PhD research. So the first step to analyze video games is to analyze actually the portrayal of archaeologists and of cultural heritage and history in video games. Here we find some problematics that derive from the figure of Indiana Jones, such as colonialism, pseudo-archaeology, and all those kind of things. So here you can see Uncharted, but also Tomb Raider that take these problems, and they have a problematic relationship with cultural heritage. Also, the representation of the past are usually reconstructive, so objective, and they are not really what's about cultural heritage and archaeology, that is interpretation and subjectivity. So the second step is to actually play video games, so validate the presence of cultural heritage in those video games and the role of the protagonist as a heritage protagonist. So here you can see one game, Pentrer, where the protagonist makes interpretations about the past. So here, Pentiment is the game. Pentiment, yeah. And so we can see some mechanics that are actually very good for archaeological interpretation of the past. The third step is to develop two video games. The first one is this war video game in the Appiantica 39 excavation project in Rome. And the second video game will be a narrative video game that will be the end of the PhD and will include all the learnings from the PhD research. The last step is the definition of new methodologies to analyze, play, and develop video games. But this is not actually the last step, because we can turn back to the first level, do it all over again, and make new methodologies and add more mechanics and more things to analyze history and see how archaeology can be depicted in a more academic way. Thank you very much.
Okay, I already have a question for you, which came out while I was listening to your presentations. And the question would be, what are the main stereotypes, the biases that you have found during your research connected to your area of research, things that are there, have been there for a long time, in a sense, creating an obstacle to a wider understanding what your sector, what your area of study can do, and how your project contributes to overcoming this bias, this stereotype. How does your project do something in terms of changing the frame of mind towards your area of research? Who wants to start? Wants to think a little bit? Good.
So for my research, I think that the main obstacles are the farmers, because I work with agriculture, and in general, farmers don't want to innovate and to utilize some sensor, etc. And so sometimes it's difficult to convince them that our technologies is more useful than the way they are to work. And so, but we are working to show them that in some day.
How do you do that? Well, I mean, how do you try and convince them?
For example, we are working really much with this azienda in Parma, and company? No, it's a farm, azienda agricola. Ah, okay, thank you. And yeah, every year we have a portion of the crop that we managed, and they have the other portion that they manage, and at the end of the season we show the results.
Okay, so very empirical, convincing them.
Yeah.
Okay. Thank you very much.
Thank you.
Very good point. You want to be next? Good. Yeah.
So my research is also about public perception. So for me, it's really important to see how people live history right now and how people connect with archaeology. I don't think that popular culture is something evil. I mean, people have some perceptions of archaeology and of history that they like a lot. They like them more than what we do or what we communicate with them. So I think that archaeology, like popular culture in archaeology and popular culture, are something that we have to take into account, because we cannot make a video game without looking what has been done and what people like. So I think that the problem are not where people think our discipline is, but some problematic aspects such as colonialism, pseudo-archaeology, and some other things that they are really problematic. So I think that we have to take those one off, but take what popular history is or popular archaeology is to do our work better, because if we do something that people doesn't like, it has no sense. So we have to think also about how people live every day with archaeology and with history and apply it to our work, doing it more academically correct, like more how we understand archaeology, but also how they understand archaeology. So,
are you doing this as part of your project?
yeah.
Mm.
Yes, this
So
is...
like a survey, or?
I'm not doing a survey because of the lack of time, because I have to also develop 2 video games. So right now is too difficult to do it like before. We have made surveys for the VR experience, and I also want to, with the VR experience, because with physical archaeology you have a really direct relationship with the past. I want to know if with the VR, with virtual reality, if this connection exists. I want to know if archaeology is just physical or if it can be made in a digital world. So yeah, we've made some interviews about the experience.
Okay, this is what I wanted to know, because there must be something that you use to evaluate, to assess what you call public perception, right?
Yeah, about this game, the first game, yes. About the second game, I will have no time, because for the end of the PhD, I want to do the demo, but I will not be able to launch it. So,
a demo, okay, good.
Thank you very much.
Then maybe for the continuation of your research.
Yeah, like to do video games, like you have to spend from 3 to 4 years, and if you are alone, perhaps more. So it's not something easy, but I think that I will do some interesting things and also methodologically interesting things.
Thank you so much.
Thank you. So in my case, I believe that the main problem is having to deal with both the industry and the market. And what I mean by that is, well, speaking about the market, I mean the public. So speaking about farmers, when you're used to a certain product, and that product works for you, it's cheap, you always go for that. Even if you put a new product in the market that maybe has the same price, most of the people are still going to go for the one that they know of. So developing a new fertilizer is not that easy in that sense, because you have to convince people that, hey, this one works too, you can actually use it, it has the same effect, but it's better for the environment. If they're used to having something else that works for them, they're most likely going to go for that. But first of all, before reaching the market, the main problem is convincing the industry that you could do something else. As I said, the fertilizers that we have at the moment in the market are basically the production process dates back to the 70s and are highly polluting, and our laws in Italy allow them to still do that. For them, it's cheap because, of course, polluting usually is cheaper. So we have to convince them that maybe spending a little bit more, but to do a product that is a little bit more sustainable, it would be better, because this is not the way to go. Like, we are polluting way.
Oh, we know that. How do we know that?
Well, that's the thing. Like, we've seen that with words, it doesn't work. So what we're trying to do at the moment is making the process cheaper compared to the main that they use at the moment, because that's the only thing that industry understands: money. I know it's bad to say, but so we're trying to make the process cheaper, to say, okay, you can invest in a new plant, but if you think about it, the process is going to be cheaper, so you're going to earn more money. And on the side of that, since we've seen that, even with that, we have been a little bit of difficulties working with the industry, we're trying to make the fertilizer itself, I'm not going to say cost zero, but as close as zero cost as possible, because we would like to use it in developing countries. So we would like to produce it and give it for free in order to support agriculture and potentially help with, world hunger. So this is what we're doing at the moment. Is it easy? Not really, but we are trying our best.
Thank you. Very, very clear, very useful.
In my case, the first difficulties, I'd say that were in the experimental part. So
I cannot hear you.
the experimental part, the beginning of the experimental session, because in our case we made all the different parts in our laboratory. So first, you have to try to understand how it works. If I made this part in this way, does it change? So the first part is, let's say, the more crafty part. And then you have to test this device first with something that is much easier to manage. So we use, for example, solutions that are similar to blood, because you have to buy blood, you have to make sure to have it. So this is also something that it's quite complicated, because it's something that you have once, and then you have to do one experimental session. But we are very lucky, because we are in the biomedical district, so we can dialogue with companies, and they also help us to set up in their laboratories. So we have the possibility to have a dialogue with them. They give us a lot of advices. So this is the lucky part, and the most difficult part will be when, if, let's say, it can become a product to sell, because it's a medical device. So you have a lot of steps to validate it, because it's something that people use, and it must not be toxic. So all the contour, let's say, and also you have to convince the medical staff that it can be very useful, because obviously hospitals already do this kind of monitoring, but it's not in the same way that we propose. So this could be in the future something very tricky to overcome, but let's say we are confident.
Thank you so much. So, any questions from the audience? I have another question. This is, I think, really tough. So it's a disclaimer, but it's our last pitch, and I think I can try this question with you. So how do you think, or how do you know, what do you know about how your research and area of research positions itself on the regional level, national level, and international level? So you're doing something, right? You're studying a certain subject, and you are aiming to achieve certain results. And have you asked yourself how your area of study, how relevant or not so relevant your area of study is, regional, national, and international? Which also has to, of course, to do with why we are here, the perception that the rest of the world, in this case Japan, but Japan as a hub for the rest of the world, so the perception the rest of the world has of Italy and of certain regions in Italy. So you see my point? It's a complicated one, but I hope you can work around it. Who wants to answer first? You do? Good.
I can start.
Yeah.
Well, in our laboratory, we have different projects that are collateral with my research topic, and these kind of projects are national projects. So, and they can be, how can I say it? They can be not crossed, but they can be
cross-fertilized.
Yeah. Okay. Yeah. Thank you. And probably, I'd say from at least at a national level, because the district is quite important from a national point of view. So if we are able to do something that can become a product, I think that it can be relevant from.
But does the project have regional roots or not?
Regional?
Regional roots, a regional source, if you see what I mean. Is it rooted in the region, and then of course it extends to the country and potentially internationally? But you see, does it happen because of the region, because of Emilia-Romagna, or could it happen everywhere else in Italy?
I think it's the first, because we are in a very specific region, in a very specific area that is developed, that has developed from scratch a context in which medical devices are the base. All people there work for that, and so it's rooted in Emilia-Romagna, yes, in Mirandola and near Mirandola, but I hope it can become something more large.
Well, this is absolutely anecdotal, but I was sharing this with my colleagues this morning. I was in Sri Lanka with my partner in August, and he was sick. He got a very serious bacterial infection, and we had to go to the hospital in Kandy, in the middle of the country, so quite difficult because it was really in the middle of the country. There was a very good university hospital, and he had to have a very strong therapy, antibiotic therapy, IV, intravenous antibiotic therapy. And then he checked, because he's a molecular biology himself, a cellular pathologist, checked the kind of antibiotic he was receiving. He came from Mirandola.
Okay.
Amazing, right? So in the middle of Sri Lanka, but that came, he read, and he also worked in Mirandola, and he said, can you imagine the antibiotic I'm receiving was made in Mirandola? So yeah, this is exactly.
I think it's
the
very beautiful. It's fantastic.
And he said it's very good. I mean, it's exactly what I was I didn't know it would be that, but it's a sort of tout ça tient. Thank you very much.
Thank you.
Okay, so as far as my project, well, it was born on an international base, meaning that it was born between a collaboration from my university, so University of Bologna, and the University of Osaka at the moment, but University of Tokushima at the beginning. So the problem itself, ivernclerosis, is international, meaning that you cannot find a single country in the world without the problem. So, of course, this is a very huge problem. And as I said, the solution, well, the aim of the solution is to bring it everywhere in the world. First of all, is to convince people that this is a better way to grow your plants, a better way to manage your fields. But it's not that easy, but yeah, I think that the problem is absolutely international. It was born between a collaboration, but at the moment we're looking to expand. So we're looking at other universities to join us in this research in order to develop the project even more and trying to find even better ways in order to make it cheaper and make it better. So yeah.
Great, thank you.
Thank you.
Love it.
So also my topic is very popular internationally because, I mean, video games are all over the world. So here in Japan is one of the most specific places in the world where video games have like this cultural importance. Also, the ones we have in Italy have a very, or should have a very cultural importance. And my project in specific is not based in Emilia-Romagna, but it's based in Rome. But in Emilia-Romagna, they are doing very interesting things with video games because there's this Italian video game program that is based in Bologna that does a lot of things with video games and culture for young people in the schools. Young people, young students. Young, like...
As in why young? No, I don't understand.
Young.
Young.
Young students in the...
Spanish and
Yeah.
I didn't say, what is young? What is a new way of defining generation? Young.
Yeah, children. So in the... Yeah, young adults. Also at Bologna, there's also the library of video games that it aims to preserve physical video games for the future. So we can see a lot of things. I think that Emilia-Romagna is a very good place also to study video games. But I mean, there are archeo gamers all over the world. So it's a topic that is under...
Archeo is really, I mean, because I know that your project is on archeo gaming. Maybe you can say a little bit more. It's a specific branch of gaming, archeo gaming, or not?
Yes, it's a very new branch of studies that comes from video game reception studies and also, of course, from archaeology. We have also historical game studies that is a little bit older and is a specific field, multidisciplinary field that studies video games, develops video games, taking into account cultural heritage, history, archaeology, and so on. It's not so strong in Italy. There are not so many archaeologists that study video games in Italy, but in Europe there are a lot, and in the United States also.
And what about in Spain?
In Spain, I've made my abroad period in Barcelona.
Okay.
Is like they really live video games. Like. Yeah? They live video games. Really? There are a lot of events, there are a lot of companies, there are a lot of startups, and there are a lot of archeo gamers, actually, that study video games and do a lot of interesting things with video games, not just for history and archaeology, but also for a lot of fields. So perhaps Italy is a little bit less... Yeah, a little bit behind because in Italy I have noticed that they have another idea about video games, that they are for children, that they are not mature, that if you play video games you are losing your time. So we have to, yeah, there's a very big bias also in academia. So we should overcome this bias and start taking seriously video games. We are starting in Italy and I hope to open the gates for other people to enter.
Thank you very much.
I think that I am more or less in the same situation of Alessia. I mean that maybe my research
is focused on Emilia-Romagna because here we have a lot of industry for food, and so agriculture is really important. For example, in Parma, the tomato cultivation are really important. But in my laboratory, there are a lot of projects that are national projects, so I think that it's more or less the same. Thank you.
Thank you very much. And if we have no questions from the floor, then I think our session is over, and I think you should join me in a very warm round of applause. Thank you. Don't go away, because the next session is upcoming.
Okay. Again, good morning to all of you. So we may start. I do see in the list Marco Fontanarosa, Paolo Gaio, okay, Sofia Tagliavini and Viola Cavallini. Enjoy the session. So I guess the first is Marco, possibly, yeah.
Can you hear me? Yeah. Okay. Good morning, everyone. I'm Marco Fontanarosa, a PhD student in chemical sciences. And today I want to show you my research project focusing on analytical chemistry applied to packaging. Plastic accompanies us in every human activity, and owing to its characteristics, has surpassed in production any other material. However, the characteristics are responsible for the serious environmental problems we face today. Global policies have implemented some measures to limit production, consumption, and disposal of plastic. And this model aligns with the 3R system, which emphasizes on reducing, reusing, and recycling resources. Both Europe and Japan promoted new alternative materials in food packaging to limit single-use plastic. However, consumers may underestimate the risks hidden in these sustainable materials due to their green concept. Food packaging is a well-known source of chemicals which are able to migrate into food and can be divided into two categories. Intentionally added substances, which refers to any substance that has been added for a technical reason during manufacturing, while NIAS, or non-intentionally added substances, corresponds to side and breakdown products and contaminants entered at any level of the supply chain. Thus, it is a challenge to identify those compounds, even if difficult and complex. So the aim of my project was the development of innovative analytical methods for identification of IAS and NIAS coming from these sustainable materials. Actually, three kinds of samples were considered: PLA Tupperware and cutlery, PET recycled bottles exposed to sunlight, and PET reusable bottles at different stages of sanitization. The idea is to combine material characterization with molecular one using mass spectrometry. Firstly, the materials were submitted to different techniques in order to get information about chemical, thermal, and surface properties, and subsequently innovative procedures were performed to maximize compounds extraction. And then an untargeted screening by GCMS was realized to allow their identification. Another crucial point is to carry out migration tests that can be used to simulate the real use conditions of these materials and to assess their safety. So, in conclusion, by combining analytical chemistry and sustainable materials, sustainability does not come at the cost of safety. Thank you very much for this opportunity.
Very good and on time. So we move on. Second presentation. Thank you.
Hi everyone, I'm Paolo Gaio, a PhD student from the University of Bologna. I'm from the Department of Interpreting and Translation, and my thesis concerns the automatic adaptation of culinary recipes. So, sorry, I always get a bit flustered. So, culinary recipes are interesting to study because they are procedural texts. So here on the left, you can see a recipe in the form of a graph where the nodes of the graph are ingredients and the edges of the graph are processes that they undergo. And what if a user maybe moves to Japan and they want to make a sort of Japanese style frittata rather than Italian style? Then you might want to substitute milk with dashi, although I was told that's not a very good substitution yesterday. But maybe if you cannot buy pancetta in Japan, you can just use pork belly or instead of zucchini, cabbage. And so on the right, you can see that a model has substituted 3 ingredients out of the graph to make a more Japanese style frittata. So this task is what is called a natural language processing task. To do this, nowadays you use state-of-the-art deep learning models. You might know ChatGPT. But there are also other ways to do it, to change a recipe. So nowadays in the machine learning space, a very big topic is thinking, is planning for language models. So as I said, a recipe is a procedural text. And so it's a series of steps. And if we want to teach, let's say in quotes, a model to think, we want it to essentially mimic the human capacity of following a plan, a long-term plan. Nowadays, with ChatGPT, you might know that if you give it, if you ask it something, it might say, "I'm thinking," and it produces a lot of text, right? That's what planning sort of is right now. But there are better ways, more sustainable ways to do it, because these models, these closed weight models, closed source models, by big tech companies are extremely expensive to run because they require thousands of processors in parallel to run them. Instead, we can use simpler models that use graph neural networks to reason over these graphs with much fewer parameters, much smaller models. And so the outcome of this project not only can help users with dietary restrictions, for example, by modifying a recipe to make it vegetarian, or maybe if you're Muslim, you can make it halal, but also we can shed some light on how planning can be done in a more sustainable way with graph neural networks. Thank you.
So good so far. So third presentation. Thank you.
Good morning, everyone. I'm Sofia Tagliavini. I'm a first-year PhD student in Health Innovative Products and Technologies at the University of Modena and Reggio Emilia. My PhD project is carried out in collaboration with the pharmaceutical company Chiesi, and it focuses on the microfluidic optimization of lipid and hybrid nanoparticles loaded with mRNA for the treatment of rare mitochondrial diseases. So to start, I would like to explain you the background of my project. Mitochondria are organelles with their own DNA, and they could be affected by PMDs and SMDs, which are primary and secondary mitochondrial diseases. Considering PMDs, they could be due to either mutation in nuclear and mitochondrial DNA, so a possible therapeutic strategy to treat them is gene therapy. This therapy faces several challenges, such as the stability of gene material in the bloodstream or safety concerns, and also industrial scalability. So the delivery of gene material inside the nanoparticles could solve many of these problems. So in my PhD project, the microfluidic technique is used as the technique to produce nanoparticles because the industrial scalability of the final product is particularly taken into account. I would use the automated nanoparticle device, which allows to push inside the chip two different phases, an organic one with inside the structural components of nanoparticles and an aqueous phase with inside mRNA. So thanks to the mixture of these phases inside the chip, nanoparticles are produced. Going deeply into my PhD project, the goal would be the development of a potential therapy for MELAS, which is a PMD, given by the mutation of mitochondrial DNA, which impact in the correct pairing between tRNA and the amino acid leucine, disrupting mitochondrial synthesis of proteins. So the idea is to deliver mRNA encoding for the enzyme leucyl-tRNA synthetase, which is this pairing. So mRNA could be transported inside cells with the nanoparticles. It would be translated in protein, and then the protein could enter inside mitochondrial where it has its therapeutic function. So this concludes my presentation. Thank Emilia Romagna for this opportunity, and thank you all for your kind attention.
Hello everyone, I'm Viola Cavallini and I'm a PhD student in physics at the University of Ferrara. During my PhD studies, I worked mostly with particles because particles are really important in the physics field, and we want to know them, to know how they behave, and we want to study them because we want to exploit particles in real-life application. We use particles in archaeometry, for example, to study old paintings and discover hidden layers, but also in medical imaging and medical physics, because we want to develop new kind of non-invasive methods to treat patients, for example, X-ray radiography, CT scans, or PET scans. But also, we want to understand particles and detect them because we want to build new dosimetry services for people that can work every day with radiations, for example, on the International Space Station, but also on planes or hospital. During my PhD, I developed the software you're seeing in the slides. It's called DataPix4, and it can help physicists understand and detect particles even better. It can configure the setup, acquire data, and it can recognize the type of particles the physicists are seeing thanks to a convolutional neural network. The DataPix4, it's used with the TimePix4. That's an ASIC developed by the MediPix4 collaboration at CERN in Geneva, Switzerland, the European Center for Nuclear Research. And it's an innovative ASIC. It has a matrix of pixels, and each pixel can detect when a charged particle interacts with it. So if we have a charged particle, like an electron, when the electron interacts with TimePix4, we can precisely know its position, its time of arrival, and the energy of the particle. TimePix4 is used with sensors because we have, like I said, X-rays beams, so photons, and we want to convert photons into electrons. So usually we use different kind of sensors to to take radiographies and CT scans, but also the main project I'm working on is the 4D photon project that you can see here on the corner. And we are developing an innovative single photon detector that it's developed in collaboration with Amamatsu Photonics here in Japan. And there's an entry window, so the photons can go inside the detector. They are converted into electrons by a photocathode and multiplied by a stack of microchannel plates. And then there's Timepics4 that detects the electrons. To conclude, there are some images on the corner of some data taking we did during those years, in particular some radiography and the first CT scan we did with Timepics4, thanks to the DataPix4 framework I've been developing. Thank you.
Well, guys, I hope you enjoyed the time you had. Going back in terms of discussion, maybe a bit challenging for you, but let's play this game. Yesterday, I discussed the fact that we hadn't innovation and inventions in the presentations. Now, instead today we have innovative as a term, innovation. Okay. What I don't see today, this is maybe the issue for discussion. Besides the first presentation, which mentioned the legislations. Again, in light of the necessity in the future to develop projects, you may, I'm sure you have the necessity to include the issue of policies, call it policies, regulations, legislations, okay? Because the word, sorry for this, the mantra in economics is market works very well, but then you say markets do fail basically always, okay? So the issue is that this is the teaching type of thing. So markets do fail, and then you have to have regulations in health, environment, education, research. So basically, it could be taxes, subsidies, standard, whatever. Okay, okay. So to finish, so you need policy regulation. So do you have in mind, yes or no, a possible regulation which could affect, be relevant now, even expected one, not just because again, maybe it's not really part of your research today, but you have, I think, to start thinking about the role of regulations and policies and legislations.
But like in terms of how it affects our field, you mean? Your
research, your field, very flexible type of reply. Yeah, just food for thought. Yeah. Yeah.
Okay. Yeah, in my sector is very affected by legislation for two reasons. The first are from in this case from Italy or Europe, we have a strict regulation about the content of recycle that has to be added into materials. So actually, companies have to follow these guidelines by the lab approach and experimental approach. Actually, EFSA, that is the agency we account for, is not set a regulation about the identification of our compounds. So we are expecting that this could be happened, but I think that it could be a long time to arrive at this point.
Yeah, so I work in AI, and nowadays I think it's probably one of the most affected. It could be one of the most affected by especially European regulation, because the EU is maybe the biggest player in this regard, because China and the US don't really care about regulating anything. If companies were actually affected and would actually comply with regulation that the EU puts out, none of them would exist, because none of them comply at all with none of the, how do you say, personal information, how do you say in English? Privacy. None of them comply with privacy regulation. So yeah, I would say definitely if for some reason they would actually be held accountable, the market would change drastically. If everyone had to play fair, the landscape would be pushed back, I would say, I don't know, 3 or 4 years from where it is now today in terms of advancements that have been made. And also, I think, maybe it's not related, but I think we're going to see quite an upheaval in the next, I want to say 18 months to 2 years, because the market is starting to realize that the whole field is a big bubble with these big techs making big promises that they overhype, and they cannot. None of this is actually as none of these models are as powerful as they portray them to be. Everyone, I don't know if the audience or the panel is aware of the talk about AGI, artificial general intelligence, but yeah, in the sector, there's a lot of talk about singularity and all of these shenanigans about humanity basically being on the brink of extinction, because none of us is going to be useful anymore to society and all of that. But I think maybe that's a bit too catastrophic. I don't know. Anyways, yeah, that's my 2 cents. Thank you.
Thank you very much. You made a good point, because maybe a bit rough, but the narrative is that Europe is regulation. US, Anglo-Saxon countries don't like regulations, more market-oriented, and then you have Asia and China, Japan, maybe more top-down. This is true, but this is true. This is food for thought. Then, for example, just sharing with you that, yes, it's true, but for example, the first environmental policy was the Clean Air Act in the US, 1972. The UK had the carbon tax and climate change levy in the 90s. So, yeah, this is true up to a point, in the sense that then life and the world is a bit more complex than Europe regulating, US not regulating, and so that's another point for your future brainstorming. But okay.
I work in a field where there are a lot of rules, because I work in the pharmaceutical sector. So before a pharmaceutical product could go on the market, there are several steps to pass through. For example, in my laboratory, we work on the formulation of nanoparticles, but we haven't the facilities to perform in vivo or in vitro before in vivo tests. So a pharmaceutical product has to pass several safety efficacy tests, both on cells models and also on animals models, because it is fundamental also this step before go on people with the disease, in order to ensure the safety, the efficacy effect of the product. So we are aware about the fact that the process before go on the market is very long, but I think that it is necessary also to ensure the safety of the final product.
I work with radiations, so there are a lot of rules, but that's that's kind of okay because we know we're safe every day. So we like those rules. Concerning the detectors, we have to, well, of course, follow the rules on safety, but mostly for us. And the only problem, we are on like the first stages of creating, developing the detectors and creating them and testing them. So we're not we do not experiment on human yet or animals. The only problem, let's say, is that when we use detectors in medical physics, but right now we're testing it on objects or pieces of biological tissues, but not on living things. So right now the only regulations is for radiation and it's for our own safety. So it's okay.
Well, it was very, very good discussion. Thanks to you four. I don't know about the time. I check with my. Are we fine with the? Yeah, the time. So I open the discussion to you there. Any kind of comments on? Let's go.
Yes, we have a lot of problems. Sorry for the switch of my language. Today, we have a big problems about also the availability of companies because searching for something that could be dangerous for them and that go against their affairs is not so positive, and it's you have to make many agreements that can make you free to publish results because you are under contract or something like that. Also, the technology is a bit difficult because the instruments we work on are often broken, and so you have to keep a constant maintenance to use it. Yeah.
So the main difficulty I have usually in my work is that it is very hard to comply with the publish or perish scheme that every researcher is subject to. Finding a research gap in a field as crowded as AI is very challenging. Usually, when I feel the despair of not seeing where to go from where I am, luckily I built a network of friends and colleagues that have more experience than me, that I rely on, and are maybe sometimes better than my supervisors at advising me. No, they're great. I'm kidding. But yeah, usually just talking it through with people that have already gone through what you have usually solves it for me. Yeah.
So my PhD project is very challenging because nanoparticles aren't new, gene therapy isn't new, but maybe the combination of gene therapy, nanoparticles, and the treatment of rare diseases is very challenging. So I try to overcome problems, searching in literature informations, or also with speaking with my professors and my colleagues, and also try a lot of different scenarios in order to find the best one. So yes, I think that yes, literature and the previous work are very useful to create a solid base for your work, but it's important also to reflect on all information and add something new and try possibilities, different possibilities to also develop something different.
I am a computer scientist, actually, in the physics department. So one of the most challenging things is to understand each other, because sometimes we just speak two different languages. But my department is full of really smart colleagues, so when I face an obstacle, usually I go to them and explain, and they can give me some real good advice. And one of the best things that I learned in the experimental physics department is that when you have a doubt or you don't know what to do, you just try it out, because it's experimental physics and you can try. So usually, yes, I do not have a role model, but maybe a lots of them, and we just try if we can, and it's really interesting. Thank you.
There is Paul. Yeah.
I think we had, great, thank you very much. We already had a little bit of chance to discuss your project. I wanted to understand the research question better. You already expressed your preoccupation in a sort of regarding the aim and the background. So maybe this is an opportunity for you, because I really appreciated your slide, but it's just that it looked like a very, I mean, you are illustrating what you're doing, but I would like to know the research question behind what you have illustrated. Thank you.
I realized it sounded more like a startup pitch or so.
Yes.
Yeah, I realized. And actually, when I wrote my research project at the start, it was even more like a startup pitch, and I switched it up a bit to make it comply with what research should be. You could think about the research question as being, is it possible to take a text and modify it in a holistic way, in a nuanced way, so that it complies with a dietary restriction of a user? And I know it's a bit a bit foggy.
A bit vague, yeah.
Yeah, it's a bit vague, but there are ways to evaluate that. You can use humans, you can use human judges, or you can use large language models nowadays as judges, or you can even just build resources, benchmark data sets. As a matter of fact, there is a data set that can be used to evaluate how good the substitutions that you carry out are. I can give you some references later if you're interested. But yeah, I realized that when I'm in the limited time I have, it sounds very, very vague, but there is some rigor under it.
No, I'm sure. So it's not. I'm not questioning the rigor. I'm questioning the communication. So I think you can do better. Can do better.
Yeah, probably. Yeah.
Yeah. So the corpus will be recipes, or this is just an example.
So the data in so, for example, if you want, I can give you some more background.
Yes, of
Usually, what you do is that you have just a text, right? You just have text, natural language. It's just characters on a screen. How do you understand anything about this? It's literally bits in ASCII format. So you need a model of natural language to extract information from this so that you can process it or reason over it. So my latest work, for example, was about extracting, improving the extraction of the entities and the processes that link them. Essentially, you can do the way you do this is that you have a neural network that takes a mathematical representation of the words, and in the output of this model, you have the prediction about a certain characteristic of a part of text or the whole text. So, for example, in the graph you saw, you could think about the full text of the recipe, which was a very small screen. There was a small screen,
yeah.
right? That had the Zafferano popular website recipe. A model would be able to find all of the nodes, the little circles, in the recipe, and then relate each node to each other node via what is called an edge, and the type of edge that connects them says what type of interaction those nodes have. So, for example, if you have a pen and egg, you could think about an edge connecting them, and then that would make a fried egg or something, right? So this was a paper that I worked on where we simply modified one operation in the model and in the output in order to reduce the amount of parameters that the model needs to carry out the task. Now that we are switching from the extraction of information from the recipe to the editing of information of the recipe, there are two ways, as I tried to mention in my presentation. You could do it the brute force way, using large language models such as ChatGPT. That's a much stupider way to do it, but it works very well because these models are amazing. They are trained on trillions and trillions and trillions of words with millions of dollars of investment. Or you could try to do it in a more nuanced way that is arguably, in my opinion, more interesting because you're using much smaller models, and so you know much better in a better way what's going on. And to do that, I intend to use what's called graph neural networks. It's basically also Viola uses, as you saw, neural networks. That's a CNN. I'm going to use a generalization of a CNN, which is called a graph neural network, which can reason over graphs. And so by using these models, you can elaborate these graphs and substitute the nodes, essentially, to carry out the intended task. Yeah.
Thank you very much. Thank you. Thank you.
Thank you for allowing me to go deeper. Thank you.
Okay, guys. So I think we can conclude on time. Just a few words.
That was very long-winded. I realized.
No, that's fine. We had a bit of time. So let me conclude with two things. First, three things. First, I really have to say that I enjoyed staying here with us. I am repeating myself, but that's true. Second, I think we have to thank Japan, the Expo, the ministry who funded Pentrer, who is funding Pentrer, the universities all together, all of you, and maybe adding, so that's really important, the region of Emilia-Romagna. So when you list things, it's something you don't have to do. When you start listing, then you fail. The last point is I would like to share something I tend to share with my team. Then maybe now they laugh, but trivial but very very relevant for your future life. I often have breakfast in Bologna, where I live, in a cafeteria whose main statement is, and then Paola could correct me, the translation in English is failing is a good thing, or making mistakes is very good, or something like that. Okay, sbagliare è giusto. To say what? To say that life is rich of failures, is failure by failure. Okay, so don't panic, and you learn. And for example, just giving you some numbers, if you have a 15% success rate in funding PEM project, you are in the top. Okay, if you have a 15% success rate, okay, this is a top figure. Okay. Average 10, low could be 3, 4, 5. Okay. It's between 5 and 15. Okay, well, when you get funding, try for submissions, applications, it's that, in between 5 and 15. So if you're very, very good or lucky or whatever, it's 15, 20. 20 means super, super, super high. So that's to say that it's something you have to have with you, because life is that. So really enjoy your career, enjoy your research, and thanks again.